Thank you very much. So thank you very much for a great introduction. I don't need to say anything else myself. And I have to say that the bar was set very high yesterday. So I hope, and I know that I have a challenging topic to talk to you about, guys. I will try and make it nice by putting lots of pictures and lots of nice, because some of the concepts are a little bit heavy. But yeah, I hope that you enjoy it. So my idea today is to give you an overview of the field of conservation genomics and to tell you how it can inform conservation actions in Procellariiformes. So we know that Procellariiformes, we all love them because they exist in all forms of forms and sizes, and they have this like amazing common denominator of an extreme adaptation to pelagic environments. But despite these unparalleled adaptations that they have, Procellariiformes are amongst the most threatened birds in the world. While only about 1/4 of, well, only, which is already quite high, 1/4 of the bird species in the world are threatened, this picture in the Procellariiformes, petrels and Procellariiformes in general, is actually more than half, which is a concerning situation. So there's lots of threats, like Procellariiformes face many threats, both at their breeding colonies in land and in their at sea as well. With the main threats being, as we saw yesterday, with lots of examples, predation by invasive species and threats at sea with bycatch wasp. But as the human population continues to increase and encroach on the natural world, we don't expect most of these threats to disappear. And actually, I don't need to convince you that there's an urgent need of effective conservation strategies to this crowd, but rather what I would like to convey to you today is how conservation strategies can benefit from the use of genomic data. That's what I'm going to try and convince you about today. But what is genomic data? Let's start with that. So because just to put everybody in the same page, and because genetics and there's several definitions of what genomic data is basically. So during this talk, I will be referring to genomic data, to genetics and genomics. So I will differentiate between genetics, which I will talk about as a small number of neutral DNA markers, typically like mitochondrial DNA or microsatellites, from genomics, which includes complete genomes or at least high-density genome-wide markers. So to convey how genomic data can assist and inform conservation strategies, I will focus on 4 topics. And for each of these 4 topics, I will give some case studies from my own research or from research from others to pass the message in a better way sometimes. So the first case will be the limitation of species and conservation units. And here we will see an example on the North Atlantic puffinus shearwaters and an example on the European stone petrel. We will also talk about identifying adaptive genetic variation. And for this, I will use an example from the Balearic and Yelkovan shearwaters. Then we will go to the assessment of inbreeding and genetic load, for which, I'm sorry, I might be also out of the club, as yesterday Nicholas was, because I will give 2 examples, one coming from the North Pacific orcas and another one coming from the kakapo in New Zealand. And finally, I will talk about genetic assignment of individuals to populations. And in this case, I will focus on the potential of this technique to link threads at sea with back to their breeding colonies, using an example of the Balearic shearwater that we are currently working on, and an example on the northern fulmar. So let's start with the limitation of species and conservation units. Speciation, as we know, is a continuous process, and speciation can be considered achieved when there's been the attainment of complete reproductive isolation. However, speciation is not deterministic, and actually it can be reverted at any moment in this process. However, like contrasting to this continuous process that speciation is, variation in nature is generally not continuous. As we can see here in this example from the birds of paradise, phenotypic diversity tends to be discontinuous in nature. However, it's normally practically difficult to have a single optimal way to divide this discontinuity into different species. And also, we have the problem that biodiversity conservation relies on the delimitation of species and conservation units to focus the conservation efforts upon. So, what is a species is a hot debate. And I will not tackle this here because we could be hours talking about different species concepts. But something that is important to mention is the general lineage concept that was coined by the Ross, and that tried to actually put the different concepts together, focusing on their commonalities rather than the things that make them different. And under these concepts, species generally can be defined as separately evolving metapopulation lineages or different branches in a phylogenetic tree. And this framework was very important because it allowed combining with genomic data and the development of multi-species coalescent species delimitation approaches, it allowed for a science-based informed species delimitation process. However, this does not take reproductive isolation into account, which we know is a very important criterion because it's a prerequisite of evolutionary lineages to maintain their integrity through space and time. So some authors argue that actually we need to incorporate reproductive isolation into species delimitation, which is challenging sometimes when we have lineages in allopatry. But in this case, combining several types of data in an integrative way, coming from genetic data and phenotypic data, can actually help us at producing a more robust framework for species delimitation. But do we need to only focus on species as units for conservation? This can have serious problems, which is the fact that if we only focus on species, we can jeopardize the maintenance of genetic diversity and evolutionary potential. Within a species, separate genetic clusters provide future evolutionary potential to adapt to changing environments, and thus there has been a growing tendency to fully consider intraspecific units for conservation. These conservation units have their goal normally is to maximize species evolutionary potential and can be used to pinpoint areas across the species range that should be prioritized for conservation. We have different types of these, and you will see in the literature there's lots of different concepts about conservation units, but generally there's two main concepts which have more or less the same purpose, but have slightly different definitions, which is the concept of evolutionary significant units and adaptive units. Evolutionary significant units are populations or groups of populations that are characterized by high genetic and ecological distinctiveness, but they don't need to be reproductively isolated. On the other hand, adaptive units are more defined about their particular genetic variation and particularly about the adaptive genetic variation that they have. So they are groups of populations that share a particular amount of adaptive genetic variation. So to put this into practice, we will focus on the North Atlantic Puffinus shearwaters. So this is a group that has been traditionally under hot debate. It includes a few endangered species, and it actually includes the Balearic shearwater, the Yelkowin shearwater, the Manx shearwater, Audubon shearwater, Boyd's, and Barolo shearwater. This group has traditionally been placed phylogenetically in different clades across the phylogeny of shearwaters. And this is due to the fact that we mainly have two main groups in here. Back. So we have this group here, which are basically small-sized shearwaters, while this group here is more like medium-sized shearwaters. And traditionally, they were placed in different groups. But using genomic data and phylogenetic analysis, we could actually show that this group constitutes a monophyletic group, which is like they have a common ancestor, which is interesting because it implies that the evolution of body size has actually been complex in the shearwaters. It's not been like a single clade of small shearwaters and a single clade of medium-sized shearwaters, but there's been medium-sized and small-sized shearwaters coming up on the phylogeny continuously. So we then focus on this group of Mediterranean and North Atlantic shearwaters, sampling across all the species ranges. And we looked at the patterns of genomic variation among and within species. And like doing first exploratory analysis using PCA, we could detect 4 main clusters, which included the Boyds and Barolo shearwater here, the Audubon shearwater here, the Mediterranean shearwaters, including both Mauretanicus and Yilkoan here, and the Manx shearwater on the top. When using also like PC3, which informs about a much lower proportion of the variance, we could also see a differentiation between the Boyd shearwater and the Barolo shearwater. So these groups, these clusters actually coincided with some of the recognized species in different bird lists. But as you can see here, the different bird lists, so bird taxonomies that are more popular nowadays, differ a lot in the number of recognized species in this clade. So to try and to harness, so harnessing the genomic data that we generated for these species, we employed an approach a multifaceted approach to define and delimit species and evolutionary significant units. And to do that, we applied several clustering-based methods, including some machine learning algorithms, some multi-species coalescence approaches, and we basically decided a criterion looking and putting all this data together with morphological data and ecological data. We decided a criteria to define species and evolutionary significant units. In the case of species, we only defined the species when there was support for more than half of these species delimitation approaches, and this support was actually, and this was further supported by ecological and morphological data. While in the case of evolutionary significant units, we considered them when there was at least some genetic differentiation and some morphological or ecological differentiation, but there was no agreement between the species delimitation methods. So these actually, on this basis, we proposed the recognition of 5 species, which is Manx shearwater, the Mediterranean shearwater, including both Balearic and Mediterranean and Yilkoan shearwater, the Audubon shearwater, Barolo shearwater, and Boyd shearwater. But also the recognition of 2 further ESUs in 2 of these clades, which is the Canary Islands population of the Manx shearwater versus the rest, and the Yilkoan and the Mauretanicus shearwaters separately. And also, like, we did not have enough data to be able to analyze the different ESUs properly in the Audubon shearwaters, so there's need for more work on that. So next we will go to the case of the stone petrels, the European stone petrels, which provide another nice example. In this case, the European stone petrel breeds across the North Atlantic, Macaronesia, and the Mediterranean. This species is considered as non-threatened by the IUCN, but actually most of the population lives in the Atlantic, with the Mediterranean population being quite small, of only around like 8,500 to 15,000 breeding pairs. And this is interesting because they have been suggested as 2 different species, as 2 different subspecies, which is on the European side, the British stone petrel, and the Mediterranean stone petrel in the Mediterranean. And these guys show some slight differences in morphology and vocalizations, with the Atlantic stone petrels being a little smaller and with shorter wings, which might actually provide them advantages to maneuver in the Atlantic with generally higher speed winds. There's very little known about its population structure, with only a work on some mitochondrial gene, which actually found some evidence for differentiation between Atlantic and Mediterranean, but it did not find any population structure within the two different basins. And also, there are some differences in the time that these guys breed. So there's some differences in there's some allochrony, although there's a lot of variability within and between colonies. So we were wondering if maybe cryptic speciation had been happening in the European stone petrel. And to answer this, we also did like a range-wide sampling for the species, including the whole Atlantic, Macaronesia, and Mediterranean, thanks to a amazing network of collaborators that actually later on became associated to work on this program called Sea Ghosts, that you can ask Banyat here in the audience or some other people involved to know more about it. And then for all these areas, we sampled DDRAT data, which is a type of genomic data that gives us around tens of thousands of markers across the genome. We also morphometrics, data on acoustics. So the genetic data actually showed more or less the same, like in the general pattern, it showed the same results that the mitochondrial data showed. So that means quite strong differentiation between the Atlantic and the Mediterranean lineages. So on the left, what we see is a phylogenetic tree of the relationships among all the sampled individuals, with the branches colored by the different localities. And on the right side, we have a mixture plot, which is a clustering method using maximum likelihood algorithms to actually assign individuals to genetic populations. But here, the picture doesn't finish here, because what we also saw from this data is that we also have 6 finer scale population structure clades, which correspond to Macaronesia, Northeast Atlantic, Cantabrian Sea, Central Mediterranean, Eastern Mediterranean, and Western Mediterranean. And also a very interesting result that we had is the genetic ancestry of the Cantabrian Sea population. So as you can see from the admixture plot, this population shows mixed ancestries, and it also shows a comb-like pattern in the phylogenetic tree. These are signs of introgression. Introgression is the exchange of genetic material through extensive hybridization. So to understand a little bit more about this, because if this introgression was recent, that means that there's no reproductive isolation between these two populations yet. So to understand that further, we did some demographic analysis using the genomic data. And this analysis actually showed us that, yeah, indeed, there had been introgression during the beginning of the diversification process of the differentiation between these two clusters. But this gene flow, this introgression, had actually stopped between Atlantic and Mediterranean populations at the recent time. So to put a little bit more of historical context to this, what we did is to complement this analysis by some analysis on demographic inference. So this plot that you can see here shows the demographic trends of the populations of the different genetic clusters from 500,000 years ago until present. Sorry, it goes this way normally in demographic inference stuff. And what we can see in this plot is that during the Holocene, there's been the population declines in nearly all the populations of stone petrels. And so what we are actually suggesting here is that this could potentially have stopped population connectivity, and thus the gene flow between Atlantic and Mediterranean would have finished at this moment. We also have like a suggestion that this could actually be caused by human arrival in different of the islands where the stone petrels were breeding, because actually the pattern at which they each of the populations falls kind of coincides with the arrival of humans to the different areas. But of course, this is a little bit speculative at the moment. So then we analyze the ecological differentiation by putting the genomics, morphometrics, and vocalizations data together. So to visualize it in a similar way, so here we have the patterns of co-ancestry between the different genetic groups, and we see the two main genetic groups characterized by higher shared co-ancestries among them, and then like really low co-ancestries between them. So when we put the vocalizations analysis in the same way, using a multivariate analysis and calculating the distances between the different genetic groups on that, we see a very similar pattern. Unfortunately, we lack data for the Cantabrian Sea for the moment here, but we can also see that the distances are between groups, within the groups than between groups. And actually, when we used the random forest classifier to classify the different individuals into Mediterranean or Atlantic, we could assign 85% of the individuals correctly. So it's a high amount of individuals that we can classify well. Then looking at the morphometrics, this is a slightly different plot. So this is also the result of a random forest classifier, but in this case for the genetic clusters. And we can see that the diagonal is the one that is more highly colored. So it means that we have more or less good assignment for individuals to their own genetic groups. But we also see that within the main genetic groups, the potentially, like the assignment is much higher within that. And also, similar to the vocalizations data, we could assign correctly 82% of the individuals. So putting this together, yeah, we have support for the delimitation of two species in the storm petrel, of the Atlantic and the Mediterranean storm petrels. And this is because we have ecological differentiation and because we show that there's reproductive isolation at the moment between the two. Then we have an additional support for ESU, which would be the Canary Islands population, which, as you can see here, for vocalizations and morphometrics, is really differentiated from the rest of the populations, and genetically as well. It generates it does a monophyletic group. So okay, so let's switch topic now and talk about something that before we couldn't do. Before the advent of genomic techniques, we couldn't actually look at this thing. While the previous topic, we could deal with it, you're looking at microsatellites and trying to understand like the genetic variation across the species. In this case, this is something new. So the identification of adaptive genetic variation basically means to identify this genetic variation that is contributing to local adaptation. So it's allowing populations to adapt to the place where they are living. So this is normally assessed using genome scans across the genome. So we normally need like high quality whole genome sequencing data for this analysis. And there are several of these genome scans that are used for that. It can be differentiation analysis like FST, genome-wide association analysis, or genotype by environment association analysis. To put an example of them, I will make it a bit nicer, just like showing some examples for each of them. So the common quail actually exists in two different kinds of ecotypes. So there's the migratory ecotype and there's the sedentary ecotype, which only lives in the south of Spain and north of Africa. And recently, looking at the differences, genetic differences between the two ecotypes, Inés Sánchez Donoso and collaborators actually saw that there was this huge peak here of differentiation in chromosome 1, while all the rest of the genome was very, not much differentiated. And this turned out to be a massive chromosomal inversion that was actually driving the differences between the two ecotypes. To change to genome-wide association analysis, in this case, I will show an example of the snowshoe hare, which inhabits North America, and actually is a species that has camouflage, and it has seasonal camouflage. So in the winter, it changes to a winter coat, which is white. But then there's some polymorphism there, and there's some populations living in areas that there's not a lot of snow cover that actually remain brown the whole year. So using genome-wide association analysis, which actually target individuals that are ancestrally close but actually differ phenotypically. Jones and collaborators actually found this, in this case, narrow region of the genome that was actually a regulator of this gene called agouti, which is known to drive the seasonal expression of the winter coat. So as you can see in these two examples, these two approaches allowed us to understand and to identify this adaptive genetic variation. And finally, there's also the genotype environment association analysis, which are widely used, and they actually, their premise is to find allele frequency changes that are associated with environmental gradients. And here we can see this example, for example, in the Elliott's laughing thrush, where in this adaptive loci for thermal tolerance, the allele frequency correlates very well with the mean diurnal temperature range. So now we'll talk a little bit more about this using the Balearic and Yelkova shearwaters as an example. So these two shearwaters are endemic to the Mediterranean and are both endangered, with the Balearic shearwater being critically endangered and the Yelkova shearwater being vulnerable. We will hear more about the Yelkova shearwater in the next talk by Dylan. And so the Balearic shearwater is restricted to the Balearic Islands, and the Yelkova shearwater occupies the rest of the Mediterranean Sea. These species differ a little bit morphologically. Actually, they can be, the morphological differences can be quite high. As you can see here, the Balearic shearwater is bigger than the Yelkova shearwater, and on average, it's darker. But for the nightmare of bird watchers living in the Mediterranean, there's a whole gradient in the Balearic shearwater that sometimes makes identification very difficult. Then they also showed contrasting migratory patterns, with the Balearic shearwater actually exiting the Mediterranean during the non-breeding season, while the Yelkova shearwater either stays in the Western Mediterranean or migrates to the Black Sea. So in the previous section, we saw that we did not find support for considering these two shearwaters as different species due to very low genetic differentiation between these two guys. However, so this actually raises the question. So we have these differences in morphology, we have these differences in behavior. So did that actually happen very fast? And this is actually what we are seeing, that there's been like a very rapid phenotypic differentiation after a recent divergence? Or it is actually that there was an ancient divergence, and that then there's been secondary contact, which has actually made the differentiation between these two shearwaters very low, despite probably at some areas of the genome it's higher, and it might be in the areas where they are actually responsible for these different phenotypes. So first of all, to understand this a little bit better, we started by doing the demographic analysis to actually answer this, using whole genome sequencing data from both Balearic and Mediterranean shearwaters. And we found that there had been like a strong bottleneck during the penultimate glacial maximum. We actually don't really trust this big number, because I will tell later why. But basically, there was a big population before that time, and during the penultimate glacial maximum, there was a strong bottleneck. Then there was a big divergence. There was a divergence during the last glacial maximum, and there's been a population expansion and secondary contact during the Holocene. So putting these results together with mitochondrial data and some other regions of the genome that we analyzed in more detail, what we actually concluded is that the evolutionary history of these shearwaters has been characterized by periods of divergence during glacial maxima and periods of extended gene flow during interglacial periods. So with this demographic history in mind, now we wanted to look at the whole genome and see if there were some areas that actually, despite the very low differentiation overall, had some peaks of differentiation. And we actually did find several, like of more or less like smaller peaks, but we found a very strong peak in chromosome 9 that we had a closer look at. So we actually looked across this area with a technique that is called local PCA, which basically looks at the ancestry and the population structure patterns across the chromosome. And the pattern that we see here is a big change of population structure in this area. And the way that this change in population structure happens in this smooth way is actually telling us that there's been a selective sweep. So there's been selection in this area happening. And this area where at the core of this selective sweep, we find several genes under this area. And actually, what is interesting is that several of these genes are involved in processes that could be involved in the different phenotypes that we see in the Balearic shearwater and the Yelkova shearwater. So some of them are involved in pre-migratory hyperphagia, which is a preparation for migration. Some of them are involved in changes in plumage darkness that could drive this coloration differentiation. And some of them are actually involved in neurological diseases that cause a motor impairment that could actually have been, genes of this sort have been shown to be involved in different migratory routes in other species. So looking at these results together, now we have also support, apart from like considering them different ESUs, they also constitute different adaptive units. So next we'll switch into the assessment of inbreeding and genetic load. So we saw that adaptive genetic variation is very important, but there are other genetic parameters that actually contribute to the ability of populations and species to persist. It's not all about adaptation. Genetic diversity as a whole is one of the three components of biodiversity recognized by the World Conservation Unit for deserving conservation. And why is that? Why do we need to conserve genetic diversity? And why has it been included in the Convention of Biological Diversity? So first of all is because genetic diversity is the raw material for evolution. Without genetic diversity, there's no evolution, and the species can adapt to their new environments. And second is because the loss of genetic diversity reduces population fitness. And we actually know from lots of empirical studies, genetic diversity tends to be a good indicator of the population fitness. So what can actually reduce genetic diversity? One of the things that can quickly reduce genetic diversity through increasing homozygosity is inbreeding. And to illustrate the role of inbreeding at reducing population fitness, I will, like the North Pacific orcas constitute a very nice example. So in the eastern North Pacific, there are several ecotypes of orcas. This map actually shows the distribution of these different ecotypes, which are the southern residents, the northern residents, the Alaska residents, the offshore, and the transient populations, which actually differ in diet, behavior, and distribution, as you can see. So within them, there are several, like some of these are actually quite genetically differentiated, while some others actually keep quite a lot of gene flow between them. And then, like following legal protection in the early 70s, most populations have actually been increasing, but not all. There's one population, the southern residents, which actually has not been increasing, and actually shows very low survival, very low fecundity. So to understand this a little bit better and to try and see like what would be the cause of this, Marty Cardos and collaborators actually sequenced the genomes of lots of these killer whales and from some of the other populations, and they actually find that the southern residents show the highest inbreeding coefficients of all these killer whales. This F-raw statistic is basically the proportion of the genome that is in segments of homozygosity that are bigger than 1 MB, than 1 megabase, so which is actually a sign of inbreeding. So to test the hypothesis of whether inbreeding might be contributing to the differences in fitness in this population, what they did is to actually compare female survival at 40 years, which is a good estimate of survival for these guys, because fecundity is more or less at a peak at their 20s, and the first age of reproduction is 10 years. And then they looked at the correlation between this female survival and the inbreeding coefficient. And they actually saw a very strong negative correlation between the two, which was a sign of a strong inbreeding depression for survival. They actually went on doing simulations with their data, and they saw that actually inbreeding depression seems to be limiting population growth at the moment. And they also actually inferred further declines if the population continues to be genetically isolated. But how does inbreeding actually affect fitness? Why is that a thing? Why does it happen? So to understand this, we have to do a little bit of theory. So this is let's imagine this is the population. In populations, there are recessive deleterious mutations that are segregating in the population. As they're deleterious, of course, never reach very high frequencies, but they stay low. They stay at a low frequency, which means that most of them, if not all, they are always in heterozygosity. And as they are recessive, being in heterozygosity, they can remain in the population. They don't cause any harm into population fitness. But what happens when, and that's what we call the masked load. But what happens when there's a bottleneck, when there's a population decline, a sudden one? So what happens then is that by genetic drift and by inbreeding, because there's less individuals in the population at that moment, mutations actually end up being in homozygosity. So this will have two possible outcomes. If they are very, very deleterious, they will be purged from the population because the individuals will die. But if they are not that deleterious, but they are actually affecting fitness, they will actually persist in the population and they will increase the load in homozygosity, which is what we know as the realized load. And it's the load that is affecting the population fitness. So this is normally the relationship that there is between these two things. And now let's come back to the orca to see if in their case that assumption from the theory it's true or not. And it is, actually. So we see that there's a strong correlation between this realized load, so the mutational load in homozygosities, compared to the inbreeding coefficient. So in this case, yeah, we have further support that inbreeding depression is actually affecting population fitness. But this relationship is not always that simple. And to see that, we will briefly see the case of the kakapo, which after an heroic recovery after the 1990s, where the bottleneck was at its highest, there's now two thriving populations of kakapo, one in South Island, in mainland, which is relatively bigger, and then a very small population in Stewart Island. So the population sizes of these two populations, we could imagine that probably Stewart Island population is under higher inbreeding. And this is actually what we see. There's much higher inbreeding in the population from Stewart Island than in the mainland. So this would also, we would also expect that then we will also have a higher amount of realized load in this population. But this is actually not the case. These two populations have exactly the same amount of realized load. Why did I confuse you here and putting this example? Well, this is just because we are starting to comprehend this stuff. We still don't know much about it. We know that there's a lot of growing examples showing that inbreeding and genetic load affect population fitness. So it is important to quantify them in natural populations, and especially in endangered populations where lots of Procellariiformes are. But we still lack a lot of the theoretical background for it. We have a simple theoretical background, and sometimes is true, but sometimes it's not. And finally, we'll move to a lighter section. I kept the lighter section for the end, just in case everybody was falling asleep already, which is the genetic assignment of individuals to populations. So Global fisheries, we know, like here, we'll hear lots of talks about this. I think it's on Wednesday. Kill millions of seabirds annually through bycatch. But seabirds, they live like as all species, as we saw before in the theory, they live as metapopulations, right? And these metapopulations, as we see in this diagram, can be more connected. The patches can be more symmetric or symmetric. There's lots of different ways a metapopulation can exist. In Procellariiformes, because they are like very mobile, we could expect that these metapopulations will be very connected, but also we know that they have a strong phylopatry, which actually makes that most of the populations of most of the metapopulations of Procellariiformes show a certain degree of population differentiation. But as they live as metapopulations, we have to keep this in mind because actually disproportionate bycatch from distinct populations can have repercussions to the whole metapopulation, and this can vary depending on this connectivity. So how can actually we link at sea threats to colonies or populations? Is that possible? Can we do that? So yeah, we can actually use genomics for that. And so one thing that we can do is that now, thanks to genomics, we know that we can find, even in species that are not very differentiated, as we saw in the case of the Balearic and the yellow Cohen shearwater, we can find areas that are highly differentiated and that allow us to actually place individuals into their own populations. So this is actually what is known as genetic stock identification. And this requires a set of source populations for which we calculate allele frequencies as a set of markers that are normally well differentiated. And then sequencing or like genotyping bycatch individuals for these same for these same SNPs, for these same genetic markers, we can actually assign these individuals to their source populations. Of course, like using some maximum likelihood or Bayesian inference methods, but this is actually possible to do. And this has actually been used to track like the main flyways across passerines, for example, in North America, where they actually sampled, for example, in this case, the Wilson's warbler across the annual range, and they linked particular stopover sites and non-breeding sites to their source populations. A little bit like if we put geolocators on the birds. So in the case of the Balearic shearwater, this species has a big problem with bycatch. And actually, from a population of roughly 3,200 pairs, around 123 individuals die annually from bycatch only in the Balearic Sea, which holds the whole breeding population of the species. And actually, bycatch accounts for nearly half of the total mortality in the species. And actually, there's been some population studies by Marie-Chase Genovart and collaborators that have shown that if the rates of bycatch don't diminish, there is a time to extinction for the Balearic shearwater of only 53 years from now. So there's clearly an urgent need to try to be able to understand and mitigate bycatch in this species, and to try and like understand if bycatch is the same for all populations or which populations might be more impacted by bycatch. We are developing a SNP panel, which is a marker panel of differentiated SNPs to be able to assign individuals from bycatch to these main breeding populations using the genomic data that we had previously. So as you have an idea of like the development of this SNP panel, this is like the PCA that we obtain with all the genome-wide SNPs, where we see that we have the power to assign each of these populations separately. And then this is the same picture with only 61 highly differentiated SNPs. And we can see that we have a very similar resolution, only with those. Here there's like millions of markers. Here there's only 61. And using this genetic stock identification, we can see that we can actually identify all the individuals perfectly, 100% support to their source populations. To see an example of this directly that has been already completed, I will show you some work from Basher and collaborators on the northern fulmar, where they tried to assign bycatch to the main breeding populations of fulmar in the North Pacific. Here, the dashed bars represent the proportion of the metapopulation that each of these populations represents. And in solid bars, we have the percentage of the bycatch. So as you can see, there are some populations where bycatch is in proportion to population size, like these two, for example. But there's others that show disproportionately high or disproportionately low bycatch. So here they provide a nice example that like some of these populations are actually really endangered by bycatch, and we can actually link specific bycatch events to populations, which can be a very useful information to mitigate bycatch. So finally, some take-home messages. So we saw that genomic data can inform the delineation of species and conservation units by providing more accurate inferences of phylogenetic relationships, population structure, and gene flow. With genomic data, we can also identify adaptive genetic variation that contributes to local adaptation and can be important for population and species persistence. Genomic data provide accurate inferences of the evolutionary and demographic histories of populations and species, allowing an evaluation of the effects of past climatic and oceanographic changes. Genomic data also permits a thorough assessment of inbreeding and genetic load, which, despite still being poorly understood, as we said, can be detrimental to population fitness. And finally, it facilitates the design of panels of genetic markers that can be inexpensively used to assign seabird bycatch to their breeding population, linking at sea threats to colonies and informing strategies to reduce the impact of bycatch. Finally, I would like to acknowledge all the people that have been involved in this work, which is a lot of people, for the North Atlantic and Pufina Shearwater, Jacob Gonzalez Solis, Jose Maria Ranz, Marta Reutor and Julio Rozas have been instrumental. For the work on the stone petrels, Federico De Pascalis, Nilo Merino Recalde, Diego Rubolini, and Jacopo Cecere have been the core group. For the Balearic and Yelkovan shearwaters, Guillem Izquierdo has been leading all the genomic work on the Balearic shearwaters, while Christian Cuevas has been leading the part on the development of the SNP chip. Marta Reutor and Julio Rozas, again, have provided supervision as well. And here is a list of all the collaborators from the different projects that I showed. I'm sure I forgot someone, but this is like all the people that made this possible and the funding agencies that made this possible too. So thank you very much, and I will be happy for questions.
You showed several examples where you were able to estimate populations in the past. I'm wondering, were some of these species that we don't know all the companies, you know where they are, like Abalis, maybe Atacama or in Caribbean, can these be used to estimate population size or tell us if there's more population spots that we know of?
Yes, but it will depend a lot on the metapopulation structure. If you have a situation where you have many populations that are completely disconnected, if you just sample the population, the source population that you know, you will probably not be able, you will be able maybe to see that there's been like a moment where there's been a divergence from another lineage. We can actually model ghost lineages into demographic modeling. So this could be species, populations that we did not sample, but the support is always lower for that. And we need like more individuals, more markers for that. But if the population is kind of like well connected, even if there's like just a few migrants per generation, then you can actually do it.
Most lawmakers and rulemakers in conservation are reliant on a largely outdated taxonomy without the benefit of the beautiful stories. How do you propose you go forward working and integrating conservation taxonomy with systematics and genetics choice?
This was clearly what I was hoping that we can actually chat and discuss here, because we were briefly discussing with Nicholas before as well. Like, of course, like this comes with some drawbacks, which is that not everybody has the resources sometimes to do this, although the prices for doing genomic analysis are really lowering at the moment. and also resources in terms of knowledge of how to analyze this data. But this is also being mitigated because we have lots of workshops. We organize workshops, for example, in Czech Republic, where we actually bring every year people that have the courses for free from places that normally they can't afford it. So I think we are going towards a place where there will be more knowledge of like how to apply this and how to actually be able to implement it. So we will see more and more of genomics being implemented into. But I agree that there's like a huge disconnection for the moment, because mostly it comes from people doing genomic analysis are evolutionary biologists normally, and lots of them are more interested in like fundamental science, like fundamental understanding of the process of evolution, and not that much on this part. But the growing of this field of conservation genomics actually provides a possibility for this interplay. And I think the way to do this is just by trying to put people like conservation biologists, like lots of you guys, together with people like me doing the work on genomics, because you can actually do the bridge between the policymakers and thing. So I the way to.
Right. We have more time for questions. I will correct that sentence. You had some wonderful examples of stuff that's going back to that data. Are there things on the horizon that you're thinking that we could learn from genetics that will just still work?
Yeah, so there's lots of development, and always like in this world, we always rely on people working on humans. So they are the ones developing all the new tools and providing like new resolution. Like now in human populations, they can actually trace back individuals to like their own places. Like they can trace back the migration of humans just by sampling you and me, for example. And this is amazing. And this is just like published last week. So there's lots of development coming, and like there's a lot of potential. Like there's a lot in genomes that we don't understand. And now we are also starting to look at not only the genetic information in terms of the nucleotides, but also about the structural variation in terms of like inversions, as we were saying. We are starting to get cheaper ways of sequencing long reads, which are like long fragments of DNA that can actually go through repetitive areas. We know that lots of the areas that we considered repetitive, and we were like, oh no, this is not important, let's just mask this part and not look at it, is actually very important, and it's most of what is driving adaptation in short times. So there's lots of, we are going to see lots of advances from like new sequencing technologies and new methodological developments that will actually open doors that we thought that were not possible. And it happens like every week in this field.
It happened. I was just curious where you catch samples at the tissue or better.
Yeah, so this is mostly the ones that we are going to start with is Jacob's samples. So Jacob has been collecting a big collection of individuals that died from bycatch, and he has them in freezers. So yeah, we are taking some tissue with a little, how is it called? Yeah, exactly. And getting some muscle tissue from them to be able to sequence those. The good thing about this SNP chip as well is that the quality of the DNA doesn't need to be very good because it's a capture method. So you just like, even if it's very degraded, it's like the fragments of DNA